your story has started when your father's sperm cell has had a close encounter with your mother's egg cell. And each one of those cells has brought 23 chromosomes. Now, the resulting cell is named the zygote. It's the very first cell of the embryo. And it has now 23 pairs of chromosomes because of what your father and mother gave to you. Now, this first cell will divide, copy its chromosomes, so that the daughter cells get the same chromosomes, and then divide again, and again, and again, and there you go. You are so cute with your very large number of cells. And each one of those cells has exactly the same genetic program, which is carried by the chromosomes. Now, to the right, you have one of those cells. In the center, there is a little gray cloud, which is the nucleus. This is the place where all the chromosomes are stored. The chromosomes are the most precious thing in your cell. So they are kept in a very safe place, which is the nucleus. Now, probably know better the chromosomes this way. So in this representation, you have two colors for each pair of chromosomes, as one which is in pink from your father, one which is in blue from your mother. So you know the sex chromosomes? If you have two X, you are a woman, X and Y, you are a man. Now you people with FSHD are most interested in one chromosome of the chromosome 4 pair, where you have this little flashlight. This is where the genetic defect of FSHD is located. Now, we zoom on the chromosomes. What are they made of? They are made of DNA. This is the beautiful DNA double helix that you see now everywhere. I saw it in advertisements for cars. Cars do not have DNA. It's only living beings that have DNA. So this beautiful double helix, discovered by Watson and Crick in 1953. Let's zoom on that. The double helix is made of 2 strands. Each strand is a very long chain that is made with 4 chemicals. You could compare these to letters. And DNA is like a language in which you have only 4 letters. And the information that is carried by DNA is only because of the order into which the letters are chained to one another. So this is named the sequence of DNA, the order of the letters. And something very special, I told you DNA is extremely precious. So if there comes an accident, some injury to one of the strands, the other strand is like a backup. When you know the sequence, so the order of the letters on one strand, you can immediately deduce the sequence of the other strand. The rule is simple. Each time you have an A on one strand, you have a T on the other one. Each time you have a C on one strand, you have a G on the other one. And so in this way, if you have too much sunshine in Las Vegas and you get some destruction of your DNA, no problem. The strand that has been injured can be repaired based on the sequence of the other strand. But that should not prevent you from putting sunscreen, okay? Now we define a gene. The gene is the recipe to make a protein. So it's a DNA segment, so a sequence of DNA that contains the information to make a protein. Okay, you will tell me now what is a protein? Well, you know when you eat a hamburger, you eat proteins. But proteins are not only in muscles. You know, hamburgers come from muscles. And proteins are all over our body. So I have no time to detail what every marvelous thing proteins are doing in our cells and our body. And I make it simpler and compare proteins to cookies. So you have them coming in all kinds of shapes and sizes and colors, and they do all kinds of things in our body, like, for instance, enzymes that are digesting what you ate for lunch. Now, things are becoming very simple. A gene is a recipe to make a protein. Expressing a gene is baking a cookie. And looking at the cell, again, you have the little gray cloud, which is the nucleus. In there is the DNA, so are the chromosomes. That's where you have the genes that carry the recipe to make a cookie. This nucleus is considered like a central library. All the books are there with all the information to make all the proteins, all the cookies in your body. But it cannot leave the library. So what happens when the cell has to make a cookie? It will copy a gene, and the molecule that is the copy of the gene is named the messenger. The messenger, or messenger RNA, is allowed to go out of the central library and go to the cell, to the bakery, where the cookie will be made based on the recipe that is carried by the messenger that was copied on the gene. Okay? So that's a very simple story. Now, let's have a look at one example of a cookie. There are all kinds of cookies. Some are even chefs. You can really understand how difficult it is to bake 3,000 different cookies in a cell. And some of these cookies are in one example, are numbered 1. The other one is to be made in 10 pieces, the other one 1,000 pieces. And so everything has to be extremely well organized. We need a bakery staff so that the cookies that have to be done at a specific time are made then and not at another time, and nobody is forgotten. Now, the cookie chef decides which cookies have to be baked in the cell, that is, which genes have to be used to make cookies. And we get a staff with a nice hierarchy, a cascade of chefs. Chef number 1 gives order to chef number 2 and 3, chef 3 gives order to chef number 4, and to nobody else, and so on. Okay? Now, I will give an example of a cookie. You are going to be shocked. 1, 2, 3. An example of a cookie chef. This is Prince Charming. Now you're all wondering, what is Prince Charming doing as a cookie chef? This lady is completely crazy. Perhaps not completely. So, we have the area where Prince Charming, the cookie chef, is working. This is at the beginning of embryo development. So you have the encounter of your father sperm cell and the mother egg cell, each one bringing chromosomes. But in fact, what happens is that the genes which are in the egg cell, so in the cell from the mother, they are asleep in the zygote. So the zygote is the first cell of the embryo. So the maternal genes are asleep, and they need to wake up, otherwise the embryo will not develop. And who wakes them up? That's Prince Charming. Prince Charming is waking up the maternal genes in the zygote to start embryo development. And this is something more realistic. Here we have to the left the zygote, so a nice picture under microscope of the first cell in the embryo. And Prince Charming wakes up the maternal gene, and then what happens? The cell wakes up, copies its chromosomes, divides in 2 cells, each one having the same set of chromosomes. This is the very first step to start making the embryo. And then those 2 cells will divide to make 4, we divide to make 8, and so on. Now, I'll tell you the truth. Prince Charming is named DUX4. This DUX4 protein you heard of, which is so toxic in your muscles, its normal function is fundamental. If there was no DUX4, there would be nobody in this room, because no baby can develop if there is no DUX4. DUX4 is really the key to going from the 1 cell to the 2 cell stage in embryo development. Okay, now FSHD is about muscle problem. So in a muscle, you have this organization with a bakery staff also. Chef number 1 gives order to chef number 2 and 3, 3 gives order to chef number 4, and so on. Now imagine, in this beautifully organized muscle cell, there comes an embryo chef. This embryo chef will start giving orders to chefs that are normally not working, that are normally asleep in a muscle cell. Like in my scheme, chef number 56, chef number 17, they normally do not work in a muscle cell. But because the embryo chef has arrived in there, started activating them, we are in trouble. Moreover, this embryo chef will not activate chef number 2 and chef number 3 that need to be active in muscle cells. So the presence of the embryo chef is screwing up all the organizations, it's chaos in the muscle. So DUX4, our Prince Charming for embryo development, has now turned into Joker. And DUX4 Joker causes muscle cell death. DUX4 is extremely toxic at every level in a cell. It will activate wrong genes, as we have seen, and then it will disturb the messengers, the messenger RNAs in the cell. It will disturb how things are functioning into the bakery. It will screw up everything, leading to cell death. Now the question is, when is this nasty joker going to be produced in muscle cells? There are two conditions. The first condition is a genetic condition, and the genetic condition is a full DUX4 gene. I'll tell you what. The genetic defect is on chromosome 4, near the end of the long arm of chromosome 4, and it is a set of repeated elements. You probably already heard that strange name, D4Z4, is the name of the repeated unit. So it's really, on DNA, something like a stuttering. Okay? So this element is repeated a large number of times. Here we have an example with this D4Z4 unit is repeated 4 times and is followed by the other DNA sequence which is on the chromosome, which is named PLAM. In every one of the D4Z4 units, there is a DUX4 gene. We have the recipe, which is shown by this green rectangle, so the recipe to make the DUX4 cookie. We have the start of the recipe, which is shown by the broken green arrow. But in none of the repeated elements is there an end. So there is no end to the recipe. So the messenger RNA that would be made from those repeated elements is not stable. It's going to be destroyed in the cell and will not be able to reach the bakery to make the DUX4 protein. Only the last unit, and this is something that was discovered in my lab, only the last unit has found an end, but look, the green rectangle is longer than in the other units. It's extending outside of the repeated elements to the PLAM region. This was completely unexpected and was found by Eugénie Anceau, a graduate student in my lab. There, outside of the repeated elements, there's an end, which is named the Belayew signal. So the Belayew signal is a trick that will allow the messenger of DUX4 to be stable, to have time to reach the bakery, and to direct production of the toxic DUX4 protein. Now, the persons who have this Belayew signal, and this is the case for all of you who are affected with FSHD, they are said to have the 4QA chromosome. So 4, chromosome 4, Q because it's the long arm of chromosome, and A because it has the Belayew signal. There are variations on the sequence of chromosome 4, and so some people have another variant, which is called another allele of chromosome 4, which is named 4QB, and these people will never develop FSHD. Okay? So that is the first condition to express the toxic DUX4 joker, is to have a full DUX4 gene with the end, so the 4QA chromosome. The second condition is an epigenetic condition. And this is an open chromatin. I'll explain what it is about. So epigenetic means around genetic. So when you look at one DNA sequence, so the order of the letters, it defines a recipe to make a protein. Epigenetic condition is not changing this order of the letters, the sequence. It's the same. It's just a little chemical modification, which is adding what I drew on this screen as a little hat, a little red hat, is going to be added onto the letter C. So each time there's a letter C, there can come a little hat. This is named a methyl group, for those of you who have done a little bit of chemistry, CH3, is coming on the Cs. And then what happens? But basically nothing, except if you have many of these little hats. DNA is said to be hypermethylated when you have many little red hats. That's on top of the slide. And when you have many little red hats, well, the cell cannot read the recipe of the gene. The gene is turned off. So there will be no messenger made, and the messenger will not direct production of the protein. If you have only a very small number of little red hats on the Cs, like at the lower part of the slide, you have DNA hypomethylation, and this allows for the gene to be copied as a messenger RNA. So then the gene is switched on. And these epigenetic changes are something that is easily altered. You can go very easily from hypermethylation, gene switched off, to hypomethylation, gene switched on. And some of you who are affected with FSHD2 probably have heard of a very difficult name to pronounce, so I will say smooshed one, SMCHD1. Smooshed one is the name of a gene that is mutated in FSHD2. So what is SMOOCH1 protein normally doing? Well, it is helping to add those little red hats on the Cs. So it is hypermethylating DNA so that genes can be switched off. And if SMOOCH1 is destroyed, then you have no hypermethylation and you have the gene switch on. Okay? So methylation, a lot of methylation, the gene is off. Small amount of methylation, the gene is on. Now we are going to look at DNA in the chromosomes. So to the right in color, you have the nice DNA double helix. And if I stretch out the DNA which is present in every chromosome of one cell, I link all those little pieces of DNA one to another, I will get from one cell a string which is higher than myself. This DNA contains all the information to make your whole cells, your whole body, and it has to fit in a chromosome that is so small that you need a microscope to see it. So we have a major packaging problem in the cell. How do I fit such a long DNA in such a small chromosome? The trick is to have help of some other proteins. These proteins are named histones. They can bind to DNA and organize them in those little bubbles that you see there, these little beads, and the little beads can come onto each other and they will make a coil, and the coil can itself supercoil, and by all this packaging story, you end up being able to fit the DNA into the chromosomes. Okay? Now, we have defined chromatin. The association of DNA with proteins is what is named chromatin. In some parts of your genome, so some parts of your chromosomes, the DNA packaging in chromatin is very, very tight. So chromatin is said to be closed. And if I take the example of the DUX4 gene, so that carries the recipe for this nasty joker, well, the DUX4 gene can be present in a part of chromatin that is very tight, that is closed. DNA will be hypermethylated, and the DUX4 gene will be off. This is what happens in the muscles of people who are not affected with FSHD. So the gene, the DUX4 gene, is present in your chromosome 4, but it is not active, it is not copied into a messenger, because it is in compact, closed chromatin. Now, in this green square, you have an area where the DUX4 gene could be located. This is a more open chromatin. The DNA can be accessed to be copied as a messenger. DNA is hypomethylated, so very few little red hats, and the DUX4 gene would be on. Now, what happens in FSHD is that there has been a switch from a compact closed chromatin that is preventing DUX4 gene expression to an open chromatin where the DUX4 gene can be active. DNA is hypomethylated, and this can occur in two ways. One way I already mentioned is to destroy the SMOOCH1 gene, which is normally involved in methylating DNA. So there will be no methylation. DNA will be in open chromatin, and DUX4 can be produced. Or in people who are affected with FSHD1, it's by reducing the number of D4Z4 copies that you get DNA hypomethylation and you open chromatin. Then what happens? genetic plus epigenetic condition, DUX4 Joker is being expressed, and it nastily says, I will destroy your muscles. Now you can realize that scientists and clinicians are trying to prevent this. And so we are trying to become like Batman. And, sorry, I went too fast. Okay, so we've been trying for many years to understand all the story I told you, plus plenty of things much more complicated, which I will not mention. And scientists are like Batman. They're trying to prevent Joker from being nasty. But Joker has been laughing for all these years, saying, ha, you have no drug to fight me. And today, at the meeting and yesterday, we had very, very exciting talks that were given by pharmaceutical companies and by researchers in universities, which are showing that we are getting really close to having many drugs. So not a single one. So many drugs are on their way, and Batman can now say to Joker, stop laughing, we'll get many drugs soon. Thank you for your attention.